All right. Welcome, everyone, to Developing Therapeutics Against Virtually Any Target with New Antibody Libraries. My name is Tim Drevens, and I'm the event manager here for Twist Bioscience. I want to just remind everyone that all lines will be muted during today's webinar. We will answer all questions after the presentation. If you do have a question, please submit in the Q&A box rather than the chat box. Following the presentation, if you have 1 minute to take a brief survey, we would greatly appreciate your feedback. You will be redirected after the webinar. And now, I'd like to introduce our speaker. Stephen Thomas is the Twist Biopharma Business Development Manager supporting Northern America. He graduated from San Diego State University with a Bachelor's of Science and a Master's in Science and Biology. He's trained as a molecular biologist and has extensive experience supporting commercial gene and variant library synthesis. Stephen initially joined Twist Bioscience's library team in 2019 as an application specialist, helping researchers worldwide identify custom library solutions to facilitate their efforts in protein, antibody, and enzyme engineering. Stephen recently joined the Twist Biopharma team, leveraging his in-depth knowledge of combinatorial libraries to support efforts in therapeutic antibody discovery and optimization. Welcome, Stephen.
Thank you very much, Tim. It's a pleasure to be here. Thank you, everybody, for attending. So it's my distinct pleasure to introduce you to Twist Biopharma today. Today, we're going to be talking about some of the fantastic libraries that we built out and how they can be used against, as the title says, virtually any target. So first, I want to begin by talking about kind of the secret sauce of Twist Biopharma, and that's basically what's on the slide here. It's our proprietary silicon-based DNA synthesis platform. So what we've done here is we've basically taken standard phosphoramidite-based chemical synthesis of oligos, and we've miniaturized it. So on this plate, which is roughly the same size as a 96-well plate that you would traditionally use for oligosynthesis, we have 9,600 wells, each with 121 unique devices, each individually capable of writing its own unique oligo. So this is really game-changing throughput, and this is at no sacrifice to oligo quality. At Twist Bioscience, our parent company, we use this to manufacture all types of really useful DNA products, so oligo pools that can be used for high-throughput CRISPR screens, genes, clonal genes at an industry-leading low list price, and then we can also use these for exome capture or target enrichment. What I think is most interesting, what we'll be talking about today, is their use in fabricating incredibly precise and high-quality DNA variant libraries. So leveraging this platform, we have significant advantages over traditional techniques for fabricating variant libraries, such as degenerate or NNK-based approaches or trimer-controlled oligos used in variant libraries. So we essentially print each and every variant that goes into our libraries, which gives us all the advantages that are listed on the screen currently. So we have base by base precision. We can use whichever codons that we want, so we can optimize for particular expression systems, or as we'll talk about, we can avoid motifs at will. These oligos don't need to be the same length, so we can incorporate length variation, which is, I'm sure is probably obvious to this audience, has its use in antibody libraries, where you oftentimes do see significant length variation in a repertoire that you're trying to mimic in your synthetic library. We can avoid sites at both the restriction enzyme, at the DNA sequence level and the amino acid level, so we can avoid restriction enzymes. So for example, if you wanted to maintain the uniqueness of a restriction enzyme site, and that motif were to come up in a library design, we could simply move to the next codon and still maintain the variant in the library. All of this is very, very precise, so we can get rates of substitution down to 1%. It's ratio controlled. We can basically introduce whatever amino acid we want at each position. This is done in a very modular format, so we can incorporate this into multiple germline scaffolds. We can mix and match heavy and light chain frameworks. And because we really hang our hat on quality at Twist Bioscience, every single library whether it's a Biopharma library or a bespoke custom library, is validated with next generation sequencing. So using oligo pools to encode for and make DNA libraries is a game-changing technique for making these libraries. These oligo pools can be designed to match the natural CDR repertoire. So instead of trying to mimic or best match the design that you're trying to screen against, we can develop libraries that exactly match the repertoire that you're wanting to interrogate. As I alluded to in the previous slide, we can completely remove all developability liabilities. Traditional knocks on synthetic libraries, basically differential post-translational modifications that you'd have in like an E. coli system versus a mammalian expression platform, we can completely remove those liabilities. So isomerization, cleavage, demidation, glycosylation sites, and so on, all of those can be completely removed from the library. We have extremely accurate representation. So again, we are not trying to mimic or model the diversity that goes into the library. We are explicitly and uniquely printing each and every variant that goes into our libraries. So the schematic that's shown on the slide here, this could be thought of, as a heavy chain of an scFv library or perhaps a VHH library. And printing out oligo pools with the diversity that's listed on the screen is basically well within our standard throughput for library fabrication. So before I dive into the biopharma libraries, I wanted to show you some data that demonstrate just how good these libraries are. So we're able to build what we call precision libraries. In this case, whether it's our design or customer design, we have a set of sequences or variants that we want to incorporate into a library specifically. So in this case, this is a heavy chain CDR3 library that has only single, double, and triple mutations from wild type for a total of 9,799 variants. On this plot that you can see, each dot represents a variant in this library. And as you can see, every single variant was represented in this library when we assessed it by NGS. Furthermore, there's remarkable uniformity with only a handful of variants on the tail, on either tail end, being over or underrepresented relative to the mean distribution. As I mentioned earlier, we can build length variation libraries either by specifically printing out the length variants that we want or by using a proprietary oligo splicing technique. And we've gotten very good at this. So we can basically build length variation libraries to exactly match the human repertoire. For naive human phage display libraries, for example, it's oftentimes useful or a desired characteristic to have a CDR3 length variation that mimics the human repertoire. With kind of middle loop length variants being overrepresented and short or long loop length variants being underrepresented relative to one another. And so you can see that's what we've done here in this case, building a CDR3 that has loop length variants from 2 to 14 residues. And you can see the design frequency of each loop length variant and the observed that we saw after we built this library are an extremely close match. So what does Twist Biopharma do? So this has been a natural extension of the products that we built out. If you kind of follow me on this journey here, Twist Bioscience originally launched by building this extremely high quality, high throughput oligosynthesis platform. We eventually expanded that into building custom high quality precision variant libraries. And then we've now extended that further, bringing in lots of industry leading expertise in antibody engineering to build out our own discovery platform here at Twist Biopharma. So essentially in the drug discovery cycle, we fit into antibody discovery and antibody optimization. As we'll get into, we have a whole suite of ready-made, pre-validated antibody display libraries for discovery. We can also assist with optimization. So this could be either affinity maturation, humanization, or to otherwise modulate for desired characteristics, ultimately leading to very potent, very specific, and human antibodies that can go into later phases of preclinical development. So on the whole, here are the key strategic areas for Twist Biopharma. As we'll get into great detail, we've built out a wide array of custom antibody display libraries. These are built on all the typical antibody library scaffolds that you would want to see. So Fab, SCIV, VHH libraries are all available with either tailored or naive diversity. We built out libraries, as I mentioned, are either naive, or we've built out some libraries that have diversity that's tailored towards specific target types. So as listed on the slide here, one of the most popular target class specific libraries we have are our GPCR antibody libraries. As I'm sure many of you are aware, GPCRs are a very sought after clinical set of class of clinical targets, but are traditionally difficult to go after with antibodies. And so we've looked to solve that problem here at Twist. Antibody optimization is another focal point. So we have lots of folks that come to us with antibodies that are perhaps derived from hybridoma techniques. And so our mouse antibodies that need to be humanized or otherwise need affinity maturation. Through all of this, we've gotten very good at producing antibodies. We have a distinct advantage in that we sit inside of a juggernaut of DNA synthesis. And so it's very easy and fast for us to quickly synthesize full-length IgGs and purify protein. So we're looking to offer that as a standalone service as well here in the near future. I wanted to really quickly touch on the high-throughput antibody production, which is our new alpha product. As I mentioned, through lots and lots of customer-driven discovery projects and internal POC projects, we got very good and efficient at expressing and purifying antibody proteins. So we decided to offer this as a standalone service as well. Eventually, this will be available on our website where you can essentially punch in your heavy and light chain sequences and receive back plates of purified antibody. So this is done in the Thermos XP293 transient system. As you can see, we do pretty extensive QC of all of these antibodies. There is a 1 or 8 mil transfection scale. So the idea here is basically to receive a low amount of lots of antibodies, which is basically what we use in-house for our discovery campaigns. Additionally, you can continue to partner with Twist Biopharma if you needed additional characterization. So we could determine binding affinity via SPR using our high-throughput Cartera LSA platform. We can do stability analyses, TM, TAG, and additional assessments of purity. If this is of interest to you, please reach out to me and I can get you in contact with the right SynBio account manager to get you set up. So this is really the vision of Twist Biopharma. So as you may be aware, there are other lots of other synthetic libraries available and companies that use those libraries to discover antibodies. However, it is traditionally very difficult, time-consuming, and expensive to build libraries to high specification. So therefore, what you typically see is companies will rely on one library that they traditionally continually go to over and over again to pan against whatever target they might be interested in. At Twist, our vision is to build out a so-called library of libraries where if a customer comes to us, we can quickly pull from hundreds of available libraries to have the best shot on goal to identify a lead antibody against their target of interest. So where one library might have a theoretical maximum diversity of 10 to the 10, taken together, we can now assess a library or a larger library of fully human antibodies of greater than 10 to the 12 diversity. So here's a snapshot of some of the libraries that are currently available. This is not exhaustive at this point, but this is a pretty good overview of what we bring to the table for antibody discovery. So we have the so-called Hyperimmune Library series. This is on a Fab scaffold, and this is based on fully human naive diversity. We also have a common light chain version of this that incorporates a fixed trastuzumab light chain framework for folks that are interested in bispecific development. As we saw in the poll, and as I've heard through various discussions with folks, is that the VHH libraries have become very popular as well, and we'll talk about this. We currently have the 4 that are listed on the screen here, which differ based on the extent of humanization of the framework and also the diversity in the CDRs. We've also built several naive libraries that are built on sort of structural-based information. So one of them that's listed on the screen here is our so-called Structural Antibody Library. This particular library is an SCFE library, which was designed based on a subset of antibodies from the protein data bank that have known crystal structures. So in this case, the theory is that because they've been crystallized, they should express and behave well and be highly developable, as any lead antibodies that come out of this library should be highly developable. And then we have a variety of target class specific libraries. As we'll get into, this diversity has been carefully tailored to bias it towards a particular target class. I'll talk in great detail about the GPCR antibody libraries 2.0 and 3.0, but the same general design premise would extend to our ion channel and carbohydrate libraries as well. So this is basically the overview of the process. It's very, very fast because we, again, are leveraging antibody libraries that are ready-made on the shelf and pre-validated, ready to go. So we begin with selections and screenings. That takes about 2 weeks. We move on to NGS and clone sequencing. So for a soluble protein target, we'll pick out roughly 100 of the top ELISA binders. For a cell-based target, where we'll use the NGS enrichment information to pull out sequence unique clones for IgG reformatting. As I've now mentioned a couple times, we can very quickly reformat these to full-length IgG or VHH FCs, as the case may be, and move on to downstream characterization. So the binding and functional assays is highly customizable, but typically will include at least an assessment of binding affinity via SPR for soluble targets. We can also do flow to determine binding for cell-based targets. And again, we run a battery test for biophysical, biochemical characterization, and we will typically do at least one cell-based functional assay as well. So the star of the show today, one of my favorite libraries, is the Hyperimmune Library series. You can see the design diversity listed here on the slide. These are built on very well-known, highly developable, highly manufacturable, tried and true frameworks, so a VH323 and a VKappa139 light chain framework. All the diversity here is fully human. So this is based on a large human database of sequences from human naive and memory B cells. What I really like to focus on here is the CDR3 diversity, which incorporates an astounding 2.5 million uniquely printed heavy chain CDR3 sequences. So again, our primary value prop at Twist is our ability to make lots and lots of DNA. So we're not using degenerate oligos to try to model this diversity. We actually printed out an oligo pool containing 2.5 million unique sequences and incorporated that into this library. So this is an incredible depth of human naive diversity that allows you to basically access the human diversity without having to use immunization approaches. We also have a common light chain version of this library with a fixed trastuzumab light chain, which might be interesting to folks who are doing bispecific development. We've incorporated length diversity into the heavy chain CDR3. So as you can see on the slide here, CDRs 1 and 2 have a fixed length. This is for the heavy chain, obviously. And then modeling the human repertoire, we have all CDR3 length variants that are seen in the naive repertoire that we're trying to match. So how does it work? Quite well, actually. So we've panned this library against many, many targets, of which I'll show you just a couple today. So one is CD3. As you may know, identifying human cyno cross-reactive antibodies is often a desired characteristic of antibodies coming out of discovery campaigns, but it's typically difficult for a CD3. And so listed on the far left here, we have an example of an antibody that was discovered straight from this Hyperimmune discovery library with pretty decent double or triple digit nanomolar affinity to the human and cyno versions of CD3, respectively. So this antibody has been further optimized using our antibody optimization platform, but this is a pretty good place to start, just coming straight out of a discovery campaign. We've also used the Hyperimmune Library for COVID-19. In this case, there are two clinically relevant targets to go after, the spike protein of the coronavirus itself, and then also the ACE2 receptor, the human ACE2 receptor, which is the entry point of the virus. So both of these are relevant targets to go after to block the interaction between the virus and the human receptor. And so this is ELISA data for two campaigns against the S1 protein and the ACE2 receptor. You can see lots and lots of hits that we got from the ELISA, lots of unique hits, which were reformatted to IgG for further characterization. Looking at the S1 data, here are some of our top leads that came out of this. This is the affinity via SPR to the S1 protein and then the S trimer, which is its natural configuration. The two listed in red have been validated as potent neutralizers of the virus. The one in green binds very tightly but does not neutralize, so is a potential candidate for diagnostic development. We have many, many more antibodies that we discovered, but these are just three of the top that we like to highlight. So shown here is basically using these, assessing some of these antibodies for their ability to block the interaction between the virus and the ACE2 receptor in a cell-based format. So here we used Vero E6 cells, which naturally express the human version of the ACE2 receptor. And we use mouse S1-RBD as a surrogate for the virus in this case. And so on the top left corner, you can see we use some control antibodies that were commercially available. So this is Acro and Abcam's S1 antibodies. And then just to the right of that, we used we're showing data for our top two leads, which have very good EC50s demonstrating potent inhibition, blocking of the virus's ability to access the ACE2 receptor. Listed down here, we have some additional leads that we've shown. So, some have good EC50s. Others, interestingly, and I won't show this data today, but others don't show good binding or blocking in this assay, but actually were still potent neutralizers of the virus in either pseudovirus or live virus assays. So moving on to the VHH libraries now. As I'm sure you know, these are very popular. So they are easy to produce and easy to engineer, typically much faster than traditional approaches. Using our libraries, of course, you don't have to rely on immunization, which is an advantage. These are small, kind of modular formats. These can be used to access epitopes that are would traditionally be sterically hindered by a standard IgG. Once discovered, these antibodies can be used as building blocks for bispecific or multi-specific antibody development. And we typically see tight antigen binding using these types of this antibody format. So on the screen here, the first two versions of the library that we built out. So the first thing to point out is that both of these are built on a consensus llama framework with naive llama diversity. And we took two different approaches to incorporate that llama diversity into the framework. So on the left is kind of a traditional ratio-based approach where we took the llama NGS repertoire and we modeled that on a substitution position by position basis, and then kind of allowing all possible combinations that, resulting to be made in the library. Another approach basically allows for only naturally occurring diversity at the CDRs. And so we call this like a CDR shuffling approach. And so that's the VHH shuffle. In that case, you can see the listed number of CDRs that were printed for each CDR. And those were allowed to combinatorially assemble one another to generate a library of 10 to the 9, 3 times 10 to the 9 diversity. So what's really cool here is we've taken this all the advantages of the VHH framework, and we've kind of moved this over into more of a human framework. So what we've done is we've made some key mutations to the llama framework. Somewhat surprisingly to me, the llama VHH antibody is not that phylogenetically distant from human heavy chain framework. So we were able to make some key mutations to the framework to partially humanize this framework, this VHH framework, which should have lower immunogenicity for any therapeutic antibodies that move into the clinic. So this is now like a DP47-like framework that we're working with here. So on the left, we have a VHH shuffle, H standing for human, which is incorporating naive llama diversity using our CDR shuffling approach. We then built a more human version of this library. So leveraging that same partially humanized DP47 framework, we also incorporated 2.5 million heavy chain CDR3 sequences into the CDR3 position. So this is the same 2.5 million CDRs from the Hyperimmune Library. So that's why this library has the HI here. So this library is basically taking advantage of both worlds here. So we get the small tight binding of a VHH framework and the extensive diversity of a naive human repertoire on a humanized llama framework. So one target that we like to go after for proof of concept is TIGIT, so clinically relevant immuno-oncology target. So this is showing you the first three versions, VHH ratio, shuffle, and H shuffle, which were each panned against TIGIT. So this is post-panning data that we're showing here, demonstrating the length of CDR3 length distributions, which kind of confirms the diversity of the library. Many of the antibodies that are shown here possess long llama CDR3s, which could potentially provide exceptional contact and binding into deep antigen channels. Sorry, did not mean to progress there. Looking at the binding affinities, so an SPR array screen demonstrated that pretty tight anti-TIGIT antibody binding affinities. So of the 141 antibodies that were measured, all had binding affinities less than 200 nanomolar, and a third of them had double-digit or better nanomolar affinity to TIGIT. So of course we use this, the one iteration of our library to go against the coronavirus spike protein. So in this case, we were able to identify lots and lots of good binders. So looking at the, so we did it using our both humanized versions of our VHH library. So the H shuffle, which incorporates fully llama diversity, and the VHH hyperimmune, which contains human diversity in the CDR3 position shuffled with llama diversity in CDRs 1 and 2. You can see a pretty impressive number of unique clones were identified in each case with unique heavy chain CDR3 sequences. So I know this is a little bit hard to see, but this is basically the output from a run on our Cartera LSA demonstrating the monovalent affinities of each of these, or a subset of these antibodies against the immobilized S1 protein in this case. So we have lots of single and double digit nanomolar affinity binders to the S1 protein. So this was a highly successful campaign and kind of, re-solidifies the advantages of using VHH antibodies with respect to the tight binding that they tend to demonstrate. So here we use these VHH antibodies in that same cell-based assay that I was referring to earlier. So again, these are mammalian cells, Vero E6 cells, which naturally express the ACE2 receptor. And in many cases, we're able to identify VHH FC leads that showed potent inhibition of binding, many more than we found using the IgG libraries and with IC50s or NC50s that rival what we saw from the IgG campaign. So kind of building on this theme, we have naive libraries and we have target class specific libraries. But among the naive libraries, we've also kind of taken a more thoughtful approach to the upfront design of some of these libraries. And one is shown on the screen here. We call it the Structural Antibody Library. And so this was built using, as a design motivation, this library was built based on antibodies from the protein data bank that have known crystal structures. So it took a while to bioinformatically mine this data and to come up with the most ideal design. Of course, we would want to we did go through and remove any developability liabilities, like glycosylation, for example, and completely remove those from the libraries. So once we settled on a set of sequences, we incorporated that again into highly developable antibody frameworks. So in this case, a heavy chain V23 and a kappa light chain 139. And you can see the diversity listed, for each CDR at each position here. Those were combinatorially assembled to build a library that, with a final transform diversity of 4 times 10 to the 10. Again, so we're kind of starting with beginning with this with a pool of sequences that, in theory, should be highly developable. But we took that one step further by completely removing any manufacturing liabilities. So this is oftentimes paired with our Hyperimmune library as a better shot on goal for an initial discovery campaign against kind of more standard targets. So as we did for Hyperimmune, we also went after CD3, again, a very relevant clinical target. In this case, we use alternating panning arms going against human and cyno homologs of this target, decreasing antigen across subsequent rounds of panning and increasing the number of washes. In this case, it was a highly successful campaign. So we were able to identify 15 human cyno cross-reactive anti-CD3 antibodies resulting from this campaign. So yeah, it's a pretty common technique that we'll use here at Twist when folks come to us for a discovery campaign to pair our naive human Fab library with one or more of our structural guided libraries to increase the likelihood of finding not only a highly specific and potent antibody, but one that is likely to be developable. Okay, so I definitely want to touch on the GPCR antibody library series. So the kind of design motivation that is shown on the screen here will apply to all of our target class specific libraries. So I want to kind of walk you through this very quickly. So this kind of came from a paper, it's listed at the bottom of this slide here by Liu et al., which basically they took a CXCR4 GPCR in complex with its natural ligand, a CVX15 protein. And they essentially asked a elegant, somewhat simple question in hindsight is, can we take the relevant binding motif from the natural ligand, graft that into an antibody, and have that antibody mimic the effects of the natural ligand? And so they did this in the context of a bovine antibody, which has an extra long heavy chain CDR3 loop, which kind of has this knot structure up at the tip. And so they replaced that knot structure with the peptide from the CVX15 protein. And they were able to demonstrate that these antibodies, these kind of motif grafted antibodies, were able to bind to CXCR4 transfected cell line. So we kind of took this basic idea and expanded this into a much larger pool of known binding partners to GPCRs. So where do all these motifs come from? So lots of things that we know bind to GPCRs. So like chemokines, helical peptides, loop peptides, the extracellular domains that send GPCRs are kind of known to interact with themselves. And of course, there are some known anti-GPCR antibodies available as well. And so we took this motif content and curated this and used this as the source of diversity to kind of bias this library towards GPCRs. But it wasn't easy. So before we could do this, we had to build an antibody scaffold that would accept all of these motifs. So we scoured our databases for all known long CDRs that we could find. and basically took out the middle and insert the motifs that we've gathered. So this was a long iterative process. So we need to figure out which CDRs and which heavy and light chain frameworks could support these grafted peptide motifs. So this basically took a year. We used a flag tag as a surrogate. And so we were basically looking for the best displayability among the various heavy and light chain and CDR3 stem combinations that we could find in the literature. We eventually settled on 2 heavy and 4 light chain frameworks and 5 different heavy chain CDR3 stems that could support these motifs in this context of phage display. So the final library includes over 100,000 motifs into the heavy chain CDR3. And these are now shuffled with naive human diversity at the other 5 CDR positions. So here's an example of a lead that came out of this, of a painting campaign using this first iteration of the GPCR library. So this is a GLP-1R GPCR receptor. So you can see on the top left here, the lead that one of the leads that we found from this campaign is that we call TB01-03. At lower concentrations, we see an inhibition of signaling. At higher concentrations, we're able to completely block the interaction, completely block binding between GLP-1R and its protein ligand GLP-1. We expanded this into an in vivo study. Obviously, we want to know if this works in the real world. In this case, we were able to demonstrate by administering this antibody to in a mouse model. We were able to demonstrate that these animals were able to maintain high glucose levels after glucose administration, which is indicative of a signaling blockade between GLP-1R and GLP-1. Of course, developability, half-life is an important characteristic for these antibody therapeutics, and we're able to demonstrate that this antibody did have a pretty good half-life in rats as well. So as kind of a complement to our motif-grafted GPCR library that I just spoke about, we also built another version of our GPCR library, this time based using the source of diversity, the design motivation, exclusively based on known GPCR antibodies. So whereas the first iteration is kind of a Frankenstein motif-grafted antibody library, this is more of a natural antibody library based on known GPCR antibodies. So we scoured the literature. We've identified 61 patented GPCR antibody sequences that target 22 different GPCR proteins. In this case, we have a mix of frameworks that basically model what we saw in the literature here. So you can see it listed on the bottom right, the frameworks that were selected and their relative representation in the library. So here's a proof of concept against a CXCR5 GPCR. So one lead that came out of this campaign, TB83107, shows good binding to hex cells that are expressing this particular receptor. And then on the right, we did a cell-based functional assay. So this is a cyclic AMP allosteric experiment, and you can see 8 or so leads that came from this campaign. We're able to demonstrate that the leads are antagonizing this particular receptor. And so you see that reflected in the shift in the curve relative to no antibody being present. So we have these 2 GPCR libraries, and they basically took different design approaches to try to tackle this problem of identifying antibodies against this difficult drug class or target class. So again, much like our structural antibody library paired with our hyperimmune, we compare these 2 libraries together to increase the likelihood of finding antibodies to this difficult to drug target class. We never stop at Twist, science never stops at Twist. So our goal is to basically add another library to our library of libraries on a monthly basis. And so we have 3 that are coming onto the scene very soon here. And these are kind of new iterations of a structural guided approach to building a naive discovery library. And so for the first one, what we're currently calling the Natural Antibody Library. So this is a library that's based on antibodies in the public patent database. So much like the structural antibody, which is based on antibodies with crystal structures, these antibodies that have been patented before are likely to be developable and express well and so on. We also partnered with some companies to build out some thoughtful libraries that are built based on some sort of rational design. So there's a company called DeepCDR that we partnered with. We built an scFv library with them. And so this is an AI augmented library that's designed based on their proprietary knowledge of DNA rearrangement and somatic hypermutation. And then we've also partnered with a company called Taurus. So as I mentioned earlier, bovines or cows have extra long heavy chain CDR3. So this is potentially advantageous for accessing different types of binding pockets. And so we're building with them a human antibody library that incorporates the advantages of a bovine heavy chain CDR3. So the last thing that I want to touch on is our optimization platform. So we call this Tau or Twist Antibody Optimization. And so what we're doing here is we are basically using your lead antibody to build a custom affinity maturation or humanization library. How do we do this? So we have a large NGS database that we draw upon to build these libraries. And so within a defined mutational space, we can basically select sequences that are nearby from your parent antibody to identify variants that will more tightly bind to the target of interest. As we always do, we will, through the course of the design, we will remove any liabilities. So isomerization, cleavage, and so on will be completely removed from the library. So if you do have an antibody that doesn't currently have these liabilities, it won't have it on after a round of optimization with Twist. And so we'll take this custom bespoke antibody library and basically pan it against your target of interest using the standard procedures, protocols that I've outlined here today. So this is very fast. Again, our specialty, our secret sauce at Twist is our ability to quickly, efficiently, with high quality manufacture DNA libraries. And so we'll take your input antibody library, your input antibody, design a library, and pan it against your target of interest and deliver back. We can identify up to 100 better binders to your target of interest. Here's some POC that came out of our campaign internally against a PD-1 antibody. So this was an antibody that on the onset had an affinity of 325 nanomolar. You can see one example of an approved antibody we found had a final affinity of 4.5 nanomolar, which was a 72-fold improvement. And this particular antibody, the data is not on the screen now, but this antibody has better affinity and better functional activity than both of the highly successful, currently commercially available anti-PD-1 antibodies. So finally, how can you work with Twist Biopharma? So we have this catalog, this library of libraries, which are available for licensing. You can license one at a time or opt for the sort of Disney Plus model and license the entire library of libraries, in which case you would continually gain access to new libraries as they became available. You can partner with us to identify leads against your target of interest, in which case we'll use those same library of libraries or a subset of them to pan against your target. So this will include all of the downstream IgG characterization, functional assessment, and so on. We're always open to optimizing leads using our Tau platform. We've identified lots of leads internally at Twist. So, I've touched on a couple of these: CD3, TIGIT, PD-1, obviously the coronavirus antibodies. All of these leads are available for licensing if these are relevant to you. Of course, we can also build custom libraries. So if you have a library design in mind that you would like us to build and pan against your target of interest, we'd be happy to do that as well. And then high-throughput IgG production. So if you have a set of antibodies that you would like to characterize, we can very quickly synthesize the DNA, transfect hex cells, and deliver an antibody product to you. So we're down to 9 minutes here. I want to be conscious of the time limitations and move on to any questions if there are some.
Great. Thanks, Stephen, for the awesome presentation. We're now going to open up for questions. So if you have a question, please make sure to ask it in the Q&A box. We do have one question, Stephen. Is your panning performed on a solid support? Are there alternate formats available?
Well, lucky for me, I have now joining us the Chief Scientific Officer of Twist Biopharma. So I know that for soluble targets, you prefer biotinylated protein, but we can do capture techniques as well. But maybe if you want to elaborate on that anymore, Aaron, that'd be.
Yeah, again, the great thing about phage display is that you can, phage can be used against all kinds of different formats of the target. So of course we can put protein down on a magnetic bead, which is our default method of doing it, but we can also pan on cells that express the antigen, and we can pan on peptides and all kinds of other forms of the antigen. So it's very flexible. So that's one of the great advantages of phage display. Next question. What are the expression levels of clones? Do these constructs express well?
Sure.
I think he's referring to the IgGs coming out of the library.
Yeah, I think that we get lots and lots of leads out of these libraries. So for a soluble protein target, for example, I know that the lab will typically do like a high throughput 3x384 well ELISA. And from there, we can pull out literally hundreds of sequences to reformat those as IgG. And so through that process, we'll be able to identify any antibodies that don't express well. And just leave those out essentially. So our platform is extremely robust in that respect, right? So if there are some leads that just don't express well, they just basically won't make it through the rounds of selection.
Yeah, just one more thing to add is, we use trastuzumab as an expression control when we make a bunch of antibodies, and almost all of our antibodies have comparable levels, maybe even better than trastuzumab. So that's a good indicator that we have good expression of antibodies coming out of the library. All right, great. Another question is, how do I pick which library is right for my project?
Sure. So we work with you on that. It's a highly collaborative approach that we use. So to everybody who's here still, we missed Aaron at the top, but Aaron is our Chief Scientific Officer for Twist Biopharma. And so working with him, working with the project manager and myself, we will help you identify the best library for your particular project. But we have kind of a set of naive libraries or libraries that are based on structural information that are workhorses for basic discovery. If you have a difficult target, which is what we specialize in, then we might use one of our target class specific libraries. So the GPCR library, for example, or the ion channel or carbohydrate libraries can be utilized to target specific target classes, if that's what you're looking for.
Great, thanks, Stephen. Let's see. Another question is, have you taken any clones and purified at large scale? What will be the expression system?
So we don't typically do large scale. It's our deliverable is a little of a lot of antibodies with characterization. But I don't know, Aaron, do you have any examples of large scale antibody production?
What we find is as we scale up, expression just gets better. So typically we'll see maybe 20-30% greater expression levels as we scale up, and we typically scale up in HEC-293, but we have done show from time to time. All right, great. Do you have any antibodies generated from rhesus, insect, or marsupial cells or soups?
That's a question for you, Aaron. I think the answer is
no. We've made, like I said, we typically make antibodies either in HEC-293 or CHO. So those are our primary cell systems. We don't have a lot of libraries from antibody diversity from those organisms. But I hope that answers your question.
Yeah, if they needed a custom antibody library with that source of diversity, then that's something that we could work with them on.
Sure. Okay. Another question, aside from your DNA synthesis capability, which can accelerate the program, what are the advantages of your program compared to other CROs that offer antibody phage display system? You mentioned that you actually synthesize all the sequence of the CDR3 region compared to others who use degenerate oligos. Is this typical what's done at other CROs?
Yeah, so other antibody discovery companies will either leverage a novel or display technique. Phage display is commonly used, and that's what we use here at Twist. And as the question alluded to, what sets us apart is our libraries. So we have libraries that are built to a specification that just can't be matched by anybody else building these types of libraries. So I like to kind of harp on the Hyperimmune Library, which has 2.5 million heavy chain CDR3 sequences. There's just nowhere that you could get that built outside of Twist. It would be very, very expensive, even if you tried to purchase that library from Twist. So you kind of get economies of scale by accessing that library. So it's that coupled with the fact that we can build these libraries to such high specification with respect to the humanization, the lack of liabilities. So traditionally in the past, there'd be like a knock on synthetic libraries in that they would contain non-desired diversity in them. Our libraries contain only wanted diversity, and it's so which increases the effective size, the number of usable variants. So you're much more likely to identify a binder because everything that's in there is fully human and is likely to be developable as well. I got to mute Aaron.
Right. One last question. Is this cost effective for getting an equivalent of a hybridoma? What I mean is, would it be nice to have tool antibodies we make in-house but we can't get or license the hybridoma or sequence?
Yeah, so definitely reach out to me and we can put together a statement of work and associated cost. It's definitely set up to be competitive with similar services out there. Definitely hybridoma has its place in the world. We're not trying to completely replace hybridoma, but the advantages that we have in terms of fully human, the potency of our antibody and the speed, is what we hang our hats on here at Twist. But I would definitely say that it's meant to be cost competitive.
Great. Thanks, Stephen, for the awesome presentation, for answering all the great questions that everybody had. Just as a reminder that following this webinar, you'll be redirected to a brief survey. And if you have 1 minute to spare, we'd really appreciate your feedback. Again, thank you again to Stephen for the awesome presentation. And thank you everyone for coming, and hope you have an awesome day.
Thanks, Aaron.